Thanks everyone for joining. I'm going to talk about the problem of managing creative chaos in scientific discoveries. I'm going to talk about 3 causes of the problem of creative chaos and also suggest 4 different ways that we can deal with it. But I'm going to start with, how do I advance the slides with this one? Got it. All right, so I'm just going to start with a little bit about myself. I'm a scientist. I have a strong passion for science. Primarily the intersection between math, computing, and natural sciences. I did some work on psoriasis disease, but also have great passion for nature. So I did some work on detecting changes on the surface of the Earth for environmental applications, the impact of coal mining. Worked with the German Space Agency. Love being on the field with my fellow scientists. But I'm also a very frustrated scientist, I must say, because there's just so many things that are slowing me down. I want to do my work. I want to do experiments. I want to try things, fail, but also make progress. So what happens is that I have this endless passion for science on one hand, but then so many things really slow me down. They're in my way. So I do what scientists do. We find a way. We have great determination and grit, and we figure things out. But the problem is it's a little bit chaotic and slow. So I'm going to talk about what exactly are the obstacles that slow me down in a few slides. But just want to say that I'm in constant mode of creative chaos, nonstop. The passion really drives my creativity. The obstacles are the ones who are creating the chaos. So I want to overcome those obstacles. I want to use my creativity, not just for overcoming the obstacles, but really focus on the science. So solving problems and challenges in science rather than solving problems in infrastructure. So here are 3 different things that are causing the creative chaos. The first one is, if I'm looking at this picture and I'm a mechanic, I'm really excited. I'm like in heaven. I'm in paradise. Look at all those parts. I can build whatever I want. I've got this great engine. I can order the different parts. And this is like the world where the research IT will be presenting these things to scientists, and they will say, this is amazing. This is amazing reference architecture. I've got all these tools that you can use. I'm providing this to you. But for me as a scientist, I really want to drive the car. I don't want to build a car every time I want to drive and get somewhere. And that's really the experience that I have as a scientist. I want to have an easy way where I can just do my work and rather than sort of building a car each and every time. So the analogy here is, think about each and every part here as Git services, Docker registries, computing infrastructure, machine learning tools, machine learning model registries, data lakes, data warehouses, databases, amazing visualization tools, and many, many more. And I need to basically stitch everything together myself in order to get my work done. So that's really the analogy, and this is the experience. And me as a scientist, I'm not a developer. I'm not someone who is very well versed with computing infrastructure and cloud computing and Linux systems. I really want, even if I code in Python and R, I'm a coding scientist. I'm not an infrastructure engineer, a DevOps engineer. So that's really my experience. I don't have the skills to do all of this. So that's the first problem. The second problem, and this is a picture taken from Central Park. You remember the COVID days? This is an actual picture, and I was there. Sort of circles. Everybody was in silos, wearing masks. And this is what happens in our organizations. Every team operates in their own silo. And because they have all these problems that I mentioned with stitching everything together and wiring it yourself, what you have is basically isolated solutions in each and every team. Each team builds their own thing to overcome those problems. And you have different solutions in different teams and different groups. And there's no way actually to reuse and share between those teams. And this is something that you're going to see in every organization. So if you are very much familiar with that in your organization, you're not alone. This is exactly what happens, working in those silos. So the reuse, collaboration within a team and across teams, it's a really big problem. The third thing is, when we get a result, we just don't know how we got the result. Imagine you're in a meeting and there's a nice PowerPoint presentation showing an amazing graph result. We found a biomarker, we found a perfect molecule, we cured cancer. And now somebody in the room is going to ask, how do we get that result? What kind of experiment did we use? What was the temperature of the fridge of all the different samples that we used? Do we know all of that? And scientists are scratching their heads. We actually don't. And now there's a problem because we really don't trust the results that we're seeing on the screen. We're excited, but we're not able to move on to manufacturing, to do validation, scientific validation, to go from research to preclinical, to clinical, to real-world evidence. We don't have a way to trace back what we've seen as a result all the way to the original data. What kind of teams were involved in this great discovery? What kind of bioinformatic pipelines? What kind of Jupyter notebooks we used? We don't know the chain of trusted science all the way to the results that we're seeing. So that's the third problem. And all these three problems really composite this creative chaos that we're living in. Now, sometimes when scientists, they're creating documentation and policies, the minute that we have some pressure to get results, we have timelines and deadlines, and we want to get there fast, everything is thrown out the window, and we just do whatever it takes. And then again, going back to the creativity and the chaos side to get that result. But if we're a biotech company, and this is, if we have biotech companies here in the audience, we need to be biopharma acquisition ready every day. Because the minute what we're incepting the company, we're already having conversations with several big pharma that they want to know what's the progress so they can snap it as quickly as possible when the time is right. But when there's a due diligence process in the biotech acquisition, the due diligence is going to kill us because we're not ready. Because the result that we were shown, it's not validated. So we came up with this concept of re-experimentation. We do the same experiment, we do the same analysis, we try to figure out what kind of version of code and data, computing infrastructure, to redo everything so that we can get the same result. And most times we're not able to get to the same result. So really, we're not biotech ready. We're not operating in a way that allows us to make progress in a very organized but creative way, be biopharma acquisition ready in every day of the week. If you're a big pharma, you want to be FDA ready. You want to have all your ducks in a row to be able to submit it. And every day that we're delayed in submission, as well as the approval, we're losing millions of dollars, really, every day. So the vision is really nothing slows science down. And how can we get to the point where we're taking these groundbreaking ideas that we have as scientists, and we want to influence the quality of our lives, our families, our friends, our communities. We want to take the brilliance of our scientists and put it into the advancement of science rather than fighting infrastructure, redoing things, and again, moving the drug discovery process from 14 years to 7 years. This is something that every big pharma is now focused on. How do you cut down the drug discovery process from 14 years to 7 and even 5 years? So nothing slows science down. That's what we want. Here are 4 ways and principles that you can implement in your own organizations to help achieve that goal. I love the Star Wars image. It's like fighting. The first thing is have a solid foundation. And when I think about solid foundation, I think about a cloud-first strategy. And there are several reasons where being a cloud-first strategy, for some people, it's going to be obvious. Of course, we're going to be, cloud-first strategy. That's clear. For other people, they'll be concerned about cloud computing costs, security, but there's a lot of research that's shown that all of these concerns can be met in a cloud strategy. And even if you have sort of on-prem and you still want to use on-prem, if you limit the use of on-prem, you'll be able to work much faster. And the reason is, when you buy hardware on-prem, it's already outdated the minute that you bought it. When you're on the cloud, you can use whatever infrastructure you want on the cloud on demand, and it keeps sort of evolving, and you get the latest and greatest all the time. So that's one thing. The other thing is, how many times have you seen sort of an on-prem cluster that is under maintenance or it's down? It happens all the time. It's always, the cloud is really giving you service 24/7, 365. There's only once in a few years that, maybe something on AWS or Google Cloud or Azure is going to have like a few issues, but then their engineers are up for the challenge and it's going to be up and ready to work in a few hours max. So starting with a cloud-first strategy, again, for some people, obvious, for some other people, not so obvious. To me, when you have a cloud-first strategy, you're much able to provide the service for scientists, as well as there's so many tools out there that are building on cloud infrastructure. So you'll be able to push the boundaries of your science much faster because you're going to be able to incorporate the tools in the cloud environment. So that's the first thing. The second thing is come up and always design and build for self-serve. You eliminate the open ticket for each and everything that you're working on. Every little thing, open a ticket, create a Docker environment. If you ask someone, this is something I've seen many times, you ask someone from Research IT, can you create this Docker image for me so that I can start doing my work? How much time is it going to take you? And the answer will be 30 minutes and I'm done. Okay, that's great. 30 minutes is great. I can wait 30 minutes. But then the person will start doing it, and then he's called for a meeting. And then he's got some other things that they need to do. So the 30 minutes are pushed to tomorrow, and tomorrow is pushed to next week because there are so many other things that they need to do and so many 30-minute tasks that these people need to do. And 30 minutes turned into a month, and I'm still waiting as a scientist. So if we can start thinking on how do we create a self-serve in everything that we do, all those small tasks, this is how we accelerate science, on these little things. This is really where there's great potential in really achieving the goal from going from 14 years to 7 years in drug discovery. The third thing is provide a centralized destination. Think about a hive when your organization is humming with activity. It's where everything comes together. You can find everything that you need. And a lot of people are talking about fair data now. Everybody knows about findable, accessible, interoperable, reusable data. But fair data is not enough. What about all the applications and pipelines? These also need to be fair. We also need to be able to find them, discover that they exist. Somebody already did like a standard for gene expression analysis. You look at your organizations, everybody has their own way of doing gene expression. And it's problematic for several reasons, because for one thing is that you can't compare apples to apples. You do one experiment and they're doing it one way, and then you do it sort of another way. Now you can't create like a meta-analysis with all the different ways of doing gene expression. So why don't we create like a gold standard way, put it in a centralized repository, and everybody can use the same approved gold standard application, pipeline, analysis, visualization. So we're not wasting our time doing the same thing, creating the same gene expression. Oh, my flavor is much better than that flavor. Why don't we agree on one, and somebody will be in charge of that. It's going to be available for everyone to reuse, and that's how we move science faster. So a centralized destination. The fourth thing is the automation part. Very much sort of comes back to the self-serve, is very much connected, but there's much more to it, because we also want to automate the tracking of everything that is happening. So it's one thing that I'm doing some self-serve with Docker, Git, wiring data, connecting everything, but there's also how do we trace all the different steps on the science? So if you have a way to automate this documentation, because we as scientists, this is too complicated for us to do. And if we have one team or one person that is not doing it properly, then we basically break the chain of science. So we can have 1,000 scientists that are doing it correctly and documenting in spreadsheets or ELNs or somewhere else. But there's enough for us to have one scientist that is not doing it in the right way, or they're rushing it. Then we already sort of broke the chain of trust. We don't know how to link the different parts in the chain of science. So automating all of this is not just a self-serve automation, but also the tracking and guaranteeing the reproducibility of each and every element, knowing the lineage. So this is my last slide. I have another one, but we at CodeOcean, we basically implemented all those principles. This is what we do with our customers, and we can help. So if you have the same problems that I was describing, we're going to be very happy to help. Our mission is really to empower scientists so that science can move faster. We're doing all this automation in the product we built. And there's a paper, white paper here, if you want to have sort of a detailed description of the different ways that you can accelerate science. You can find it here. And you can also find us in our booth in 308, right in the EXO. Thanks so much.